Good afternoon, everyone. Guess we'll begin. My name is Jeff. I'm a current graduate student here in Boston, and for the last year, I have been moderating the communications working group. So I'm happy today to be presenting not only our strategic roadmap and charter, but also the achievements of our working group over the past year. So one thing which hopefully you knew before you even walked in today is that GP Write has been showing up in the news. We have had a tremendous reception in the press over the last 6 to 9 months, including cover stories at Newsweek as well as Wired Magazine. And recently there have been articles in Chemistry World and Neolife. So I think it is due to a bit of a congratulations for the people involved, not only involved in making those articles happen, but about GP Write generally for the really positive press that we've received over the last year. So please join me in congratulating. That is, again, in marked contrast to this sort of beginning of the project, and I think we've really sort of left that legacy behind. And part of the way that we're really defining our message is, as I sort of alluded to this morning, not only reaching out to the media to tell our story, but also creating our own content and making sure that the story gets out there. So as you already know, I'm tremendously proud to unveil these scientific infographics. So there are 2 of them, one of which is about GP Write itself and about what the project is, who's involved, what we're trying to do or trying to accomplish, and sort of the sorts of problems that you could imagine solving via the proposal of GP Write. And additionally, specifically for this meeting, we have put together an infographic that explains the virus-proof cell line. What is recoding and why is this something that a member of the public should care about? So again, this is sort of in line with this 2-pronged approach of not only reaching out to the media and making our presence known and our achievements known through that avenue, but also telling our own story and making sure that gets put out there. So with that, I'll sort of delve into the strategic vision that has been guiding those efforts and will continue to guide future efforts. So I've sort of distilled our group charter and roadmap into a few slides. To begin, what's the mission? So the mission of this communications team is really quite simple. It's merely to get the world as excited about GP Write as they were about putting a person on the moon. So that, I think, should be easily accomplishable. In order to do that, there's a few things that need to be sort of baked into all the content that we produce. That includes these operating principles like transparency, scientific accuracy, clarity and simplicity, as well as making sure that people see their values reflected in the sort of work that we do. And this is something that I mentioned in my remarks last year. Additionally, there's some sort of key messaging that we've been focusing on. One is, what is GP Write and what is the potential impact on human health and well-being. Second is this commitment to transparency. As sort of mentioned this morning, the LC group is going to be tremendously important in this aspect, and on the communications working group, we really see as part of our mission to facilitate that public debate and make sure that public debate is meaningful, because I think we have in the past suffered the consequences of not really going out there and making sure people know what's going on. So this is very important going forward and something that we're already putting into action. Who's involved? This is an open international consortium with lots of different stakeholders. And finally, the GP Write is enabled in a lot of ways by the organizational capacity of the Center of Excellence for Engineering Biology and coordinating these efforts and making this meeting possible and for sort of putting this organization on the best course. And then in terms of who we're producing content for, that would include the scientifically engaged public as well as the science news media. The thinking being that these are groups that are most naturally going to be tuned into what we're doing and perhaps interested and supportive of the project goals, and that if we can really cultivate a strong relationship with these constituencies, we can use that to branch out into the broader public that may not fall into these camps. So what does that, that's the 30,000-foot view. What does that translate to on a day-to-day basis? So first of all, that includes coordinating media engagement, which we've done a very large amount of in the lead up to this meeting. I kind of want to throw a few things out to you as members of the audience. So, is your lab or working group or organization producing something that, the world should know about? Or sort of on the other hand, are reporters seeking you out or are asking you questions? In the same way that GP Wright scientifically, by coming together, we're more than the sum of our parts. I think the same can be true of our public communications and outreach. So if we coordinate these, how can we really boost each other's signal and maximize the impact of the limited resources that we have? In terms of signal boosting, we can also think about external partnerships. So one great example is, every university is producing press releases and materials to support their scientists and the discoveries of those scientists. So can we work together? Can we share that content or provide content that the universities can then distribute to their network so that we can really, again, kind of use the resources we already have and deploy them in the most effective way. And aside from academia, there's tons of potential for reaching out to, for example, museums, community science organizations, disease foundations might be very interested in the work that we're doing. On social media, in just the past month, we've been able to reach a tremendous number of people, and especially with all the news we've generated today, this number will undoubtedly be much higher. So this is one of the tasks of our committee. Again, I sort of mentioned this already, but how can we better connect with the people who are interested? So, for example, we could think about making a newsletter every quarter and sending that out to even just participants in the working groups to keep people abreast, so that we don't have to convene once a year to figure out what each other are doing. And then last, obviously this will never happen, but in the case of a crisis, that sort of falls under our committee's purview. And this is sort of the same content, essentially, I was just presenting, but in table form. So these are the sort of goals for the working group in the near and midterm. Looking over these as I was sort of putting this together, I thought about how these are all very important, but they're also a bit obvious and they're not very adventurous. And so for a project that started out talking about making photosynthetic humans or bomb-sniffing plants, how can the communications team produce something that lives up to the ambitions of the project generally? And so I was sort of thinking about this, and I was drawn to this quote that was included in the article that showed up in Wired magazine. So this professor from BU is talking about when he thinks of writing genomes, I like to think of the different genres people could write. Personally, I like fiction, coming up with these sort of fantastical creatures. So this, to me, made me think of a game. And games, scientific games have definitely, they have precedent in research. So for example, this game or this application Fold It came out in 2010 to help predict protein folding, and the public was very receptive. There have been over a dozen publications and many more than that in terms of mentions in the media. Maybe a closer analogy to GP Wright would be the game Spore. So it's like a sandbox type game that I played when I was in high school, where you sort of design a creature from scratch and then let it loose in this sort of in nature, quote unquote, and let it sort of interact with its environment and with other organisms and sort of evolve. So I wonder if we could produce a similar game for GP Wright, where someone could, in a sandbox style, produce their own genome on their computer and then let it loose in the wild. So you could think of it as like SimCity for your genome, as someone put it out. So again, so my point in bringing this up is not to say, this specific idea is the idea that we're going to move forward with, but just to plant this seed that, hey, for such an ambitious project, let's think big in terms of public communications. With that, I have the very important task of acknowledging all the work that went into this. So aside from myself, these are all the members on our working group. Special thanks to Nancy for her guidance for our committee. Carol Miller, you may have seen her outside. She's very busy and has been for the last month organizing all the media outreach and the media relations for this meeting, so she deserves a tremendous amount of credit for her work. She was helped by Lynn and also Amy. Has also been very helpful. And then the rest of the members of the group have obviously been crucial towards everything I was able to present today. And with that, I'll add a quick plug to communicate with us. I've been heartened so far that basically everyone else who has come and spoken has expressed interest in interactions between the working groups. So especially since our role is to boost the profile of GP Wright and the goings-on. It's especially important that we interact with you, be it, joint phone calls or increasing our networks in between working groups. We welcome new members, especially for producing new content. We really want to build out this capability. Put us in touch with your university or your organization. We've already spoken, for example, with Yale, and they were very enthusiastic about working with us. And then again, connect and share on social media. We've been tweeting and posting all day, so be sure to check that out. I think next what I'll do is welcome up Ellen from the education working group, and I think the idea was that she would present and then we would have a joint panel between the two working groups. So with that, I will temporarily step aside.
I'm obviously with the Education Working Group, and we are one of the smallest groups. It turns out educators are very busy, and particularly at this time of year, the end of the semester, we couldn't get anyone else to come to the meeting. I think that the education group spent a lot of time agonizing over involvement in GP Wright, which may seem kind of silly, but I think that they felt that whatever we were doing and whatever content we were producing, we didn't want to seem like we were advertising or advocating anything, that we wanted to look for things that would truly add to what's already out there in DNA science education. Because in some ways, this project isn't so different from a lot of other things that are happening and that already have some sort of educational components around them. So we wanted to make sure that there was something unique that we could bring to education from this particular project. So our mission, we worked on obviously a charter and a mission statement like the other groups, is if we wanted to think big, what would be near and dear to our hearts was that everyone would have a familiarity with DNA science. I think we've all heard the comments like, I would never eat a genetically modified tomato, there's DNA in it, and things like that. And also that the information that we conveyed was accurate, particularly about expectations. We would like to see more of a respect for this type of technology. It seems like it's been battered around in the news in so many different ways, but the kind of respect for science that I remember having when I was a kid and we were looking at the moonshots doesn't seem to be around anymore, and we'd like to bring that back. Just an awareness that this stuff it's embedded in your daily life. You're all wearing genetically modified cotton clothing. It's in half the foods you eat. It's in the medicines that you take. It's in the rubber in your tires and the plastic in your products. It's everywhere. And so the general public really doesn't have a feel for that. So all of a sudden the discussion became a lot bigger than just the GP Wright project, and something that we were all very passionate about. And also the understanding that GP Wright is part of this and has something very special to give in terms of it being this large moonshot project that has a great deal of potential to do good. So our guiding principles was that we would try to present everything as accurately as possible. And also, my committee was very impassioned about things like embedding ethics and societal contexts. You can have an ethical discussion with a kid that's like 5 years old if you're talking about, you know, capturing an ant and studying it, and is this fair to the poor ant? And it can open a whole discussion around animal experimentation and everything else, and you'd be surprised at how sophisticated these discussions can get. So embedding that into all of the lessons was something that everyone was very, very much on board with, and we are going to be working closely with the other committees in GP Wright to make sure that that happens. Also inviting the students to be part of the conversation, forums, discussion, stuff like that. These are things that really a school with absolutely no equipment and no lab facilities can get their students excited about. And then also trying to make the information accessible to all levels, not just like the AP Biology superstar private school kids. Really drilling down. All of us on the committee work with inner city kids, and we really, really want to see more diversity in the sciences. And the last thing, which is sort of a no-brainer, is don't reinvent the wheel, because there's a lot of good programs out there already. So what our group does is we spend time, first of all, defining the scope of what, because the scope could be enormous. It could be everything from just sticking to sort of what the group members know best, which is sort of around the high school level, which is when people actually get DNA in the regular curriculum, to extending it down to the K through 6 level and up through higher levels of education, and also potentially to the general public in terms of lifelong learning and informal science education, which is sort of what I'm famous for. So. We sort of started on really big and then had to drill down a little bit to try and figure out what we were going to do first. So obviously, we would be designing curricula. We would have to have metrics for it. I mean, all of these goals are pretty obvious. This is the current sad state of science education in the United States. The PISA study in 2015 also said that we were, I think, 18th in the world in science education. There's a lot of good existing programs, and what we decided to do as a first strategy was to build on these. You can add modules to these. These are all sort of modular learning things. And so I think the low-hanging fruit for us was to first start by adding some existing modules to these programs around the technologies and concepts of GP Wright. At the high school level first, because that's what this committee is most comfortable with, but if we expand the committee to include people who are educators at other levels, we would be happy to have discussions about other levels. And then make sure that they were widely accessible. We have access to some pretty amazing web-based resources, like Rockefeller University's new web platform for disseminating this type of information. And as the GP Wright project progresses, we would like to hear from all of you. If you have an aspect of what you do in your lab that you think that you could turn into a cool module, we would really, really like to hear from you. And we look forward to trying to have more interaction with the actual scientists that are doing this. And you may be running educational programs in your own schools. I know Jeff had an amazingly successful Build a Genome project that he ran for years at the college level. And then eventually it would be nice to actually hold a competition. The Genes in Space program is huge. Every high school teacher is talking about it. I don't know what the equivalent would be for this, but I'm trying, and hopefully we will find something equivalent that will generate some more excitement around the project in schools. And of course, we'd love to go international, and I'd love to talk to people who are doing this project in other countries and see what resources are available, because right now, I am only familiar with the United States. This was one thing we came up with. So this was inspired by a conversation with Leslie Mitchell about the dark matter in chromosomes and how we don't know sometimes what parts are controlling what. And so the idea of saying we've got modular parts, there's a structure to eukaryotic genes, which is somewhat mysterious. Sometimes we don't know what exactly is controlling it. It may be 100 base pairs upstream. It might be 100,000 base pairs upstream. And you could do a simple lab exercise around this where you could have a couple of pieces that you switched out to see which one actually controlled the gene. So obviously a lot of these things in these modules, even the ones that exist now like the DNA fingerprinting, they don't actually do DNA fingerprinting. They do some sort of facsimile, like sort of a replica of it. Another thing, and please, I am not an artist, be kind to me, was I think ultra-safe cell, the word ultra-safe is not going to appeal to any high school student. So I was thinking maybe supercell, and that there could be some sort of imagination, and this is sort of like the game that was just presented of sort of what could you make a cell do, what would it be possible to make a cell do if you could really get in there and rearrange chromosomes and do whatever you needed to do to make something happen. And then just the idea of large-scale synthesis, where you would take modular pieces and fit them together. And maybe this could be something like a simple Gibson assembly exercise or something, just to say that you can actually take a bunch of different pieces, throw them into a test tube, and my goodness, you get something that actually works. Anyway, any ideas would be greatly appreciated. We just submitted, you may not be aware, but we just submitted our first project proposal around this. And we also hope to interface with some of the other great organizations that are starting public conversations about this. I just was at this Editing Evolution Forum that George spoke at that was around public discussion about genome editing. But you could see some of the same questions might come up in people's minds looking at this project. And it was a really useful exercise and a lot of fun, and definitely something that we'd like to incorporate into the education space. Thanks.
Great, yeah, I guess we'll take some questions now. We have a few minutes.
I have a question. Who here is involved in, as well as research, in an educational program of some sort around your work? So quite a few of you. If you could give information about that to our committee, that would be really helpful. How would they do that, Ellen? How would they do that? How would they do that? They could write to me, or at Biotech Without Borders, or any of the other committee members, really. I don't have a Twitter handle for it at the moment. I'm behind the communications.
Why don't you ask them to submit their ideas to info@engineeringbiologycenter.org?
Okay. Did everyone hear that? Submit your ideas or your data or your description of your programs to info@engineeringbiology.org? info@engineeringbiologycenter.org. Okay.
I guess I have a question as well. So sort of now that we have these roadmaps put together and some momentum, I imagine it'll be a bit easier to approach other working groups about, specific ways that we could interface. But I wonder, maybe to the other working group chairs, what do you think would be the most effective way of, for example, getting your findings over to the communications or the education team to create content based around that? Is it going to be harder to create a genome or to get a robust conversation going between the working group chairs? I strongly hope it's the former.
Scientists hate working with the media.
That's why we'll do it for you. Just, we'll let us be the middleman.
Well, you're exceptional in many ways.
Sure, absolutely. And so, yeah, any educational module or communication we work out, we definitely need to strongly consider. Regarding the games, in the case of Spore, you are creating an organism that then evolves, which I guess is seems a little oxymoronic because you design a thing and then send it out, which, there's its own mixed messages. But I think that I like the sandbox aspect of, here are a bunch of, here's an eye and an ear, and, kind of put them together in this Frankensteinish genome. That aspect I like. I'm not sure if you would, maybe then instead of sending it out into the world like you do in Spore, you would grow it in a test tube or on a petri dish and sort of see what the properties are. And
It's
maybe
kind
you're
of like the supercell idea, in that you would try to get people to imagine sort of the happy side of being able to give a cell attributes. Kind of like with the Human Genome Project that George has. You're always saying that people contain superpowers that they would be like to give to someone else. Like if you have an unusual immunity for something, or some other thing that might be useful for other people to have and to share it. If you can imagine something like that within a cell being created, what would you like to share through the cell with everyone, rather than let's see what we can create so it can stomp around and.
Right. Yeah. So the analogy with sport doesn't hold perfectly, but I think the broader point of let's be very careful about what we do. I definitely hear.
On the other hand, it's interesting. There's a game, I think it's called Plague. I can't remember. I've only played it a couple of times. But essentially, when you play it, you try to start an outbreak, and you try to get the plague to win. And it's actually very difficult, because if you make something, it gives people an understanding, is if you make something virulent enough, it kills off everyone before it can spread. And if it manifests, then people start creating a cure for it. And so in a weird way, it kind of shows you how futile it would be to try and create something, because in 99% of the cases, it can't go anywhere possibly. But I don't think we want to go there.
Yeah, Carolyn.
I was suggesting it as a module, not necessarily to change the name of what we're doing. And I'd like to say there's a certain spillover between our committees, in that for those of you who don't know me, I started the first community lab in the United States, and probably the world, I think. But this network of community labs are places where people can be citizen scientists, and they can come in and learn from people who know what they're doing about these sorts of things, and actually practice them hands-on. And they're great ways to get scientists and the public to communicate with each other in sort of this neutral ground. It's not like this big scary lab in an institution. It's a local neighborhood place. The equipment is usually kind of shabby and used, and it's a very comfortable atmosphere to learn this stuff in, and it lowers the barrier to access in terms of being able to understand this kind of stuff and not be afraid of it. Because there's nothing worse than feeling like somebody is doing some highly sophisticated technology that's affecting your life, you don't understand it, and you can't control it. So I think that goes a long way for diffusing that. And if you look at what these labs do first, they usually take exercises that they can buy in a kit form from Ward Scientific, Carolina Biological, all of these companies that sell kits for high schools. And so the stuff that we create for high schools is eminently scalable out to the general public in terms of public workshops in these informal science spaces. So there is a lot of sort of overlap between our committees in that sense.
Yeah, and we've had a joint, well, at least one joint working group call to discuss the overlap, because that's kind of why we're presenting together right now, I think, as well. As there's a few questions. Yeah.
Hi, here. Hi, my name is Binay. I'm from India. I'm actually involved in the high school biotech curriculum development in India. I think one of the problems that we face in most of the high schools, the kits are very expensive because many of our schools don't have expensive equipment. It will be very useful if you can come up with a kit or the suggestion where the high school students can do experiments to learn about GP Write, and both in terms of learning about the technology using videos and then doing a small experiment. And the experiment can be done across the world just by using I think the key word is not using expensive equipment, because many of our schools don't have access to equipment.
That's a fabulous point. And, ethical discussions will only go so far to assuage that sort of hunger for being involved in science. So one of the things that's very exciting about the way technology is going is you can get this sort of remote stuff. If you look at a company like Transcriptic, that basically their business model is you type what you want us to do into a computer and some robots somewhere else do it. I would love to have, if nothing else, at least a contest where students could propose some sort of change to a genome, and it could be done by a GP Write scientist. Or, maybe there could be a whole small program going where we could actually let students do remote builds and show them the results. I think that would be a great idea.
Yeah, I think one of the reasons that at least we find in our country that a lot of high school students, very talented one, not interested in biology because they think that it's only for the rich labs. And they see this big equipment, multi-million-dollar huge labs. So I think what we need to do is to create a consensus among the public that biology can also be done with less expensive, a little less money. Around
Also,
the world.
the reading and writing of DNA code, the price keeps coming down. So, for example, to do the cheek swab mitochondrial DNA analysis, it's $3 to have it sequenced, Sanger sequencing. I think we're.
Do we have time for one question, or are we done? All right. This gentleman.
Is to launch, for example, maybe it's possible to launch a contest of writing a novel according to certain rules on GP Write now, or maybe in 5 years, 10 years, 20 years, 100 years from now, and the outcome of what it could be for undergrad students up to older people, and potentially having several prizes, and the one that would win the prize maybe would have support to write a book after this. And with certain rules, it means it has to be extremely accurate scientifically, possible as scientifically, and it could also impact people that are not only scientist-oriented, but scientifically oriented, but maybe with a broader potential, and could be fairly educational.
I think in some sense the Center for Excellence is already sort of doing that. They have some project proposals that are from citizen scientists that are being evaluated, so it's broadly accepting of ideas.
All right, thank you very much.